Where does all of that good structural information come from? A couple of techniques, and we can break it down like this. If we had a time machine and went back 10 or 15 years, virtually all of the known structures come from X-ray crystallography, like this. And that's a technique that works well on any protein that you can get to form a crystal. And so the crystal will be that protein aligned with another copy of that protein, forming a thing called a unit cell. And they'll have other molecules of water, or you can add different agents. Basically, there's no one recipe for how to make a crystal. And most proteins are not crystallized and maybe cannot be crystallized. So we had in the database a lot of crystals of a few very easy to solve things, relatively easy to solve things. And for most things, we had just no crystal structures. But over time, we've had a couple other methods come along. NMR, nuclear magnetic resonance, really good technique, but it's very difficult to fully solve proteins that are longer than, maybe 300 amino acids. It's already getting dicey. You might be able to get to 500, but yeah, you won't like how you get there. And larger proteins are just too difficult to solve. But the neat thing with X-ray crystallography, it gives you one static view of the crystal, usually a dried out version of the protein. It's like looking at a mummy and trying to imagine what the person was like. You got to kind of rehydrate it and imagine it moving around a little bit. NMR will give you a range of structures. So they'll normally bundle together the 10 most likely structures, with the best evidence, and they'll show the protein as it's moving around, as it's doing different things. And so those are quite interesting when you can get one, when it's amenable. And then a lot of the recent structures, and probably the fastest growing field, is cryo-electron microscopy to reconstruct it, which is basically looking at it, taking a picture, stacking thousands of those pictures up together, and then making a go of it. I'll show you X-ray crystallography and cryo-EM reconstruction, which is something I'm more familiar with here, and there are other techniques that exist, and that's fine. We can't cover everything. So, without further ado, X-ray crystallography. So yeah, it's like high-resolution microscopy, except in a really indirect way. So you've got an electron beam shown here as this little white arrow going through, and the electron beam is passing through a crystal. So all the proteins are organized so that they're facing exactly the same way as the protein above and below, right, left, in every direction from it. And in that X-ray beam, all of the X-rays that come out are just about exactly parallel. So when a bunch of parallel X-rays hit a bunch of things that are in a static organization, you'll get particular deflections. Like every once in a while, some of these X-rays are going to get flicked, backwards or deflected a little bit from where they were headed. And essentially, every amino acid, every atom in there is going to deflect some X-rays. And so, if you can reconstruct it, if you can reconstruct the little dots that in the pattern, using the hidden dimension, which is called phase, then you can solve a crystal structure and you can get a very nice molecular representation of what the protein looks like. This is basically how we understand proteins. So yeah, X-rays are diffracted by crystals. That's the basics. Fine. Uniform light, meaning that you have X-rays of the same wavelength and traveling in just precisely the same direction. They can do this by just having an X-ray source and either focusing the X-ray or just you can just block everything that's not going directly straight with like a little pinhole. And so, yeah, they're going to hit the molecule, and it's going to be a crystal of proteins. You say, protein crystal? That's weird. But yeah, you can get it to happen. It doesn't happen very readily. It doesn't happen for most proteins. So in order to get a crystal, you first have to have an almost completely pure protein. And then you have to get lucky. That protein has to like to stack together like little Lego bricks. And it has to be happy doing that in a crystal. And most proteins, nearly all proteins, do not behave like crystals when they're actually doing their job. So essentially, none of these proteins signed up for this. None of them was naturally selected for its ability to form crystals. And so you're trying to get things to crystallize that never don't know what a crystal is, essentially. Why X-rays? Well, duh, yeah. The wavelength's small enough. Light has these wavelengths of 300 nanometers, so it's this giant thing. And remember, average atom, or like distance between two atoms, is something like 1 angstrom, so that's 1/10 of a nanometer. So if your wave is coming through every 300 nanometers, most of the time you're going to miss that particular atom, and you'll have to put a ton of waves through to occasionally hit it. And even then, you'll only know it was somewhere within that 300 nanometer area. And a protein is much smaller than 300 nanometers. Like a big protein will be, I don't know, 50 or 100 nanometers. So you couldn't do any better than yes or no, I ran into a protein. With light. Electrons, about 10 nanometer wavelength, but they do some interesting things. And you can actually, if you have enough data from these electrons, you can actually reconstruct molecular models down to 2 or 3 angstrom resolution with the best techniques. And that's really something. Yeah. And so that's nice because it's a different way of doing this. It's not dependent on the crystal. The protein can be floating around in water that you freeze as ice, but they're in sort of random orientations, basically doing what they were doing at the moment before you froze them. Yeah, really nice. Anyway, X-rays are teeny tiny. They're about 1 angstrom in size. And so on a good X-ray reconstruction, you can get down to 2 angstrom resolution. Yeah, that happens with the best structures. Most of them end up being in the 2 to 3 or 3.5 angstrom resolution range. Bad electron microscope structures range from, sort of a little higher than that, maybe 5 to 15 angstroms. Really bad ones can go up to like many nanometers. They still tell you something. It's just not very high resolution something. And light is fine for finding cells and maybe organelles and not much of anything smaller. So there you go. Okay. So X-rays can pass through, which is refraction. X-rays can be deflected, which is reflection. Rather, it can sort of bounce off. Or X-rays can diffract, which is this crazy wave property. And it's this diffraction pattern that essentially we're producing and that we're going to rely on. And X-rays pretty much don't care. They'll go through anything. Yeah. Or most anything. Yeah. So it was invented by various people. They got various Nobel Prizes. Protein crystals are like little chunks. Yeah, they look like little jewels. And the crystal shape, you'll sometimes have multiple crystal shapes in the same preparation, which actually can reflect sometimes different organization of the proteins. Because just like you can stack cannonballs in different ways and still make a little cannonball tower, you can do the same thing with crystals. Actually, it's a lot more complex. It's more like the way you can stack Legos in different ways. The X-ray beam will actually burn and incinerate the crystal. It will destroy the crystal as it is creating the diffraction pattern. So you can collect some data from a crystal, but you actually need lots and lots of these little crystals if you want to solve an entire structure. So one crystal will usually not do it for you because you're not just going to record it with the crystal in one orientation. You're then going to rotate the crystal and take another shot, rotate it again, rotate it this way, so that you have lots of different views of this, which just gives you more information on what's next to what. And then the diffraction pattern of the crystal is basically, you can basically reconstruct what one unit of that crystal, one asymmetric unit, like one protein or clump of proteins, looks like, where all the atoms are in there if you do it right. So it's pretty neat. Yeah. You do that in what's called Fourier space, the Fourier transformation, which is essentially what, it's a way of simulating what's happening between the interaction of the X-rays and the atoms. Why do you need a crystal? Obvious, right? You need everything aligned the same because you'll have many different X-rays traveling through, but they'll all be going in the same direction. And so you want them to, you get a brighter spot, if you get more diffraction, and you get more diffraction if you have, more targets, and you're basically shooting more arrows. That's all it is. So yeah, a crystal is well ordered. And sometimes what you'll see is that only parts of the crystal will be well ordered and other parts will be a little bit floppy. In that case, you can only solve the parts of the crystal that are actually stationary, basically well ordered and consistent. The parts that differ within the same molecule within the unit cell, those generally can't be reconstructed. So back in the day, 85% of structures were from X-ray crystallography. That number is declining, and cryo-EM is coming in, but it's still an X-ray crystallographer's world out there. The thing is, as an X-ray crystallographer, you may work 5 years to get one structure. And it may be an important structure, it may make your name and your career, but the 4.5 years leading up to that are a very uncertain time, and it's very expensive. The current rates, last I checked, it was about $100,000 per crystal structure in time, in materials, in people, in X-ray source rental, all this stuff. So, okay. So yeah, the computer is going to reconstruct that using a mathematical equation called a Fourier transform, which will essentially, it has the property, it's more complicated than this, but it has the property of organizing things by relative distance. So things that are far apart from each other, like from one molecule to the next, will appear right around the center. And little tiny changes, like the position of a single atom, these will be very far out from the center. So you kind of draw a circle around where your good data is, and then you figure out what's the last sort of shell, circle, in which you have data, and how complete is that data. And then those are some of the statistics that let you say, all right, we have resolution out to, this much data, and it's 98% complete there. But if we were to go one step further, it would be 5% complete. It's just like a cliff. Okay. So you purify the protein, you crystallize the protein, which often involves making smaller seed crystals that you then enlarge. You're then going to, usually in modern crystallography, you'll probably have an in-house X-ray source where you can check to see if your crystals diffract. Because a lot of crystals don't diffract, or they turn out to be crystals of the salts and other agents that were in the buffer. One of the common methods is still used is the hanging drop method, where you put a very protein-rich drop on the underside of a slide, and you just let it hang there and slowly evaporate, hoping that as the water goes out, the proteins come together and make that little crystalline organization. Then you'll go out and collect data, which means shooting X-rays at it and melting your crystals until they're gone. Then you will try to solve the structure, which is a process called phasing. And there's some things you can do, like you can load in extra large atoms. Selenomethionine is something you can load in, which has a great big selenium atom on it. And it just makes, instead of these little tiny spots, you'll have a gigantic black spot wherever it's bouncing off of one of those selenomethionines. And so that helps. That helps to solve the phase component. Like if you imagine, a two-dimensional plot, the phase, you can think of it kind of like the third dimension that's there. It's an additional dimension that you can't see, but that you totally need in order to understand what the heck is going on. And then once you have the phase and all of these, you're going to do structure determination, which means basically finding a structure model that will fit the available data that you have and still obey the laws of, the way proteins fold to some extent. And so most things that diffract and where you can phase it, you can almost always solve those. Getting it to crystallize, very error prone. You'll get maybe 5% of things to crystallize. Of the ones that crystallize, only some of those will diffract meaningfully, like well, in a way that you can solve it. So now you're down to something like 1% of the proteins you try will actually come out the other end and you'll be able to solve it. So it's pretty brutal work, but I'm glad people do it because they're just the best pictures in biology. So protein purification. How do you do it? Quite often with a tag. So a tag is a little piece of a protein that you attach to your protein. And sometimes you can have a tag that has a little protease cleavage site on it, so you can cut it off later so you don't have to have it there. But a lot of the time people will just solve the structure with the tag present. Because basically, if you can get a structure, it's a really good day, week, year. Yeah. And you'll take it. But there's different things that you can try in that. And so with a tag on there, you can then do affinity purification. For example, if you have a thing called a histidine tag, which is 6 histidine amino acids in a row, those will wrap around metal ions. So they'll wrap around cobalt or zinc ions, for example. And so you can have a column that has immobilized metal ions on it. And so all your proteins will stick to the little immobilized cobalts or zincs on there. And then you can put in another chemical, something like imidazole, and wash them all off. And then you can try to refold them just by taking away the denaturant you put on. And who knows? You might get a purified protein. This is the stage where most proteins fail. They either don't express well or they do not purify well. Okay. And then the often most difficult step after this, not the first step, but is to get an adequate crystal. Because only certain types of crystals, there are certain crystal orientations that are absolutely useless for X-ray crystallography. You need kind of a nice three-dimensional array where you've got asymmetric units packed in all the different dimensions. There are a couple kinds that work well and a few other kinds that are very common and are utterly useless. And so then you can put it in a little goniometer, a little thing that can rotate it just so. And you shoot X-rays at it and you collect the X-rays on a little screen on the other side. That's it. You're X-raying the little crystal. So your crystal needs to be itty bitty, like usually less than a millimeter, which is good. It's hard to make bigger protein crystals. And it has to be absolutely perfect. No little air bubbles in there, no little cracks, no little planes where it kind of is one crystal here and another crystal over here. You'll see that with like salt crystals and like at gem and mineral shows. Twinning and stuff happens all the time. You may be able to occasionally split those apart, but usually, your goose is cooked. Yeah. Data collection. Synchrotron is an X-ray source, which will just be a large sort of circular thing that will then, you can sort of channel the X-rays off in different directions. They'll have different what they call beam lines from this. That's a typical way to do it. They'll shoot through the crystal and you'll have a little detector on the other end. And the crystal will be attached to a little stand that's sort of mounted on that you can rotate it around, and so you can see it from different angles. That's the idea. So that is roughly what a diffraction pattern can look like. And so all the information toward the center is about the packing of The proteins information out toward the edge is of sort of the orientation of individual atoms. And if you Fourier transform that, you can get an electron density map of the crystal. This is the idea. And so that's fine. And then from that, you can begin to try to refine your structure. And so there's our map, and we needed to phase this in order to solve it this far. And there's the structure of the thing we were trying to solve, and so there you go. It's a good match. The lines in here are like a topographical map. Yeah. So they're if they're closer together, this is like, it's like height. So imagine these as like little hills, little bumps. It's just little sort of mounds of electron density, which is what atoms kind of are. Yeah, because that's where the electrons are to be found. Okay, uses. Yeah, you can solve the structure of pretty much anything that you crystallize. Let's just say that. Yeah, biomolecule, chemical, whatever. Yeah, it's very nice. If you want to see exactly what you have in a sample, all you have to do is purify that unknown, crystallize it, and solve the crystal structure. No big deal. And so yes, this is useful because it can be used to even solve very large macromolecular protein structures. And I guess it's involved in dairy science. I'm not going to read that. But yeah, you need a robust sample. It has to be able to stand up to a little bit of X-rays without changing shape or, something like that. It should be clear, no sort of cloudy or inclusions or air bubbles. Twinned samples where you got two crystals growing out from a common point are difficult to handle. You have to try and position it so you're only shooting through one of the twins at a time. And even then, yeah, good luck. And yeah, data collection often requires, you normally send up 2 or 3 people and they'll work in shifts over a couple of days to get as much beam time as possible for the travel and dollars and rental of the beam line. So there you go. That's how you do it efficiently. This has been X-ray crystallography.